Hi, Michelle Glass here. Welcome back to another episode in Chapter 10 lecture series. Our topic for this video is just a basic review of nucleic acid structure. So our whole topic for Chapter 10 is looking at the DNA molecule and how it works in the cell. And so we need to make sure that we go back and revisit the basic structure of our DNA molecule and also our RNA molecule. Now, the nucleic acid structure, specifically DNA, structure was discovered and described in 1953. So pretty fast after we figured out that DNA was the hereditary molecule. Now, very often Watson and Crick are receiving the big credit for the discovery and characterization of DNA structure, but they could not have discovered it without the help of Rosalind Franklin. So she's actually credited for producing an X-ray of the DNA molecule itself, which was critical to Watson and Crick being able to understand and then describe the nucleic acid structure. So it's a pretty interesting story. So remember that our deoxyribose nucleic acid is double-stranded structure, and we're going to see that the backbone of this molecule is consisting of a 5-carbon sugar and a phosphate molecule. So we can describe it as a sugar-phosphate backbone. And then connecting the two sides together, you have nitrogenous bases. We have 4 possible bases. These are typically abbreviated with the first letter. So we are seeing here adenine, thymine, cytosine, and guanine. And they fit together, connecting the two sides in a very specific pattern. And we describe this as complementary base pairing. So adenine will always pair with thymine, as you can see, and cytosine will always pair with guanine, as you can see. And there's a fun little memory device. You can pick either one that you like. Apples grow on trees, so A always pairs with T. That's my favorite because I like apples and I like trees. But you could also pick the car is in the garage. So C, cytosine always pairs with G. The other thing that we need to pay attention to is that our double helix is actually antiparallel. And what we mean here is when we look at our 5-carbon sugar, you can see that I've drawn that in for you here, each one of those points in the molecule is where a carbon fits in. And just to simplify the structure, we don't write in those carbons. So you can actually see, of course, our 5-carbon sugar has 5 carbons, and we start by counting them and assigning them as a 1', 2', 3', 4', and a 5'. Do you see how those are labeled? Okay, so what we're going to see on one side that we have our 5' end up, so we're going to talk about it as going from 5 to 3', and on the opposite end is going in the opposite direction or orientation, so it's going to have the 5' end pointing down. Take a look at this picture here. Notice then that the phosphate group is going to be attached to the 5' end, and the hydroxide group is attached to the 3' end there. So we will see in the 5 to 3' direction that the phosphate group is pointing up and the hydroxide group is pointing down, and we see the opposite of that on the other side. This is going to matter when we talk about DNA replication, so we'll come back to this idea in the future. All right, and we can see that kind of added to this particular diagram here. So we're going to see that one backbone is in the 5 to 3 direction, and the other piece is going in the 3 to 5. Again, you know, really those previous drawings were just to help us see how we're getting that nomenclature. Key thing we need to remember is just that they're going in opposite directions, and we call that antiparallel. Okay, just quick reminder, we have another nucleic acid that will be important in our conversations in Chapter 10. It is called RNA, which stands for ribose nucleic acid. Now, this is a single-stranded molecule, so it's going to be smaller, but a lot of the structure is the same. It still has a sugar-phosphate backbone, but it has a different 5-carbon sugar than deoxyribose nucleic acid, and that's how we're getting our different names. So RNA is named for its sugar, which is ribose. We're still going to have nitrogenous bases. We still have 4, but we do see that there's a difference here. So we're going to have adenine, uracil, cytosine, and guanine. So it's important to remember RNA molecule has uracil, but no thymine. And that's it for now.